If you count the number of bacteria in culture over some time, this is the graph you get. You have probably seen the time divisions as lag phase, log phase, stationary phase, and the decay phase. From this plot, you see that the max number of bacteria is found in the stasis phase. When expressing proteins from vectors, we care about a healthy protein yield. So you never want your bacteria culture to enter a decay phase. Going from a few cells to a crazy large number, our job is to maximize the protein yield. There are many factors you can change to maximize the yield, but more often than not, the choice of promoter and its regulation contributes most significantly to the yields than the rest. You could have your protein expression vector express your protein without any control. These are the constitutive or always active promoters. A promoter from glucose metabolism gene would fall into this category. Should you choose to express your protein without any control, which means from time 0, the bacteria starts the protein expression. This will result in the formation of two types of bacteria. One slow-growing, which represents a small fraction of the population, and one fast-growing, which will represent a higher percentage. Unfortunately, these slow-growing bacteria are the ones that still express your protein, and the fast-grower ones, even though they have your expression vector, fail to express the protein. Now, if we were to say that 1 unit of protein equals 1 bacteria, and our aim is to get maximum yield, unfortunately from a system without any control, your yield is only 10%. The 10% is not factual, I'm just using it as an example. If the protein was a little bit toxic, then this number would be 0. In the other case, imagine you could control the activity of the promoter, and you let the cells grow and grow until they reach the max density. So in the stasis phase, you induce the expression. In contrast to the constitutive promoters, these will be inducible promoters, meaning that they are off by default but can be turned on when you want. With such a control, all bacteria will grow happily, so upon induction, you expect to maximize the protein yield. In the case of constitutive expression, the slow-growing cells express your protein, and the happy ones have found a way to not express your protein. They're happier and grow faster, and lose the protein expression because they have evolved by accumulating some mutations. In the case of induction, everyone stays happy while dividing with no mutations. Let's go a step further. At a molecular level, the bacteria is happy when it can fulfill its metabolic demands, like transcription and translation. In constitutive protein expression, the bacteria has a lot more transcription and translation to do, and suddenly there are not enough ribosomes, not enough polymerases, not enough ingredients to cook. This increases cellular burden and stress. As more protein is made from the vector, the cells also get congested, which can be toxic. These bacteria therefore divide slowly and live a sad life. Because of this stress, some bacteria naturally try to evolve and accumulate mutations in the vector such that it loses the protein expression. They mutate towards happiness and overtake the sad bacteria in numbers. In case of induction, the only thing a bacteria has to do is to divide. Till the point of induction, they live happily because the metabolic burden is not strong. So, how do you build an inducible protein expression vector? Let's start with something simple. Here's an example, where you can control the expression of your protein using a promoter and operator from the lactose operon. If you put this vector in a bacteria, you expect the LacI to make the repressor, which binds to the operators on the plasmid and blocks the RNA polymerase from transcribing the ORF in the front. Unfortunately, there's a minor problem. In a typical bacteria, there are only about 10 or so molecules of LacI at any given time made from one LacI gene in the genome. Given that you can have about 50 or so copies of your vector in the bacteria, 10 LacI molecules are not enough to inactivate 50 operators. You could argue to engineer and artificially add more LacI genes into the genome, but a better way to do this is to add LacI gene in the vector itself, and that solves the problem. Now, if you want to activate the expression of your protein in this bacteria, as you would in stationary phase, you just add IPTG to the culture. IPTG binds the inhibitor, making it non-functional, resulting in mRNA and then protein production. However, these types of promoters all suffer from leakiness. This is called basal expression. Most inducible promoters, such as those derived from LacP operon, like TAC, which is a hybrid of tryptophan and LacP operon, and LacUV35, which is a mutated version of the LacP promoter that makes it independent of CAP region. All these promoters depend on negative regulation, meaning that LacI must inhibit the operator. If at any point the inhibitor fails, the protein will be expressed. In contrast, ARA-bad promoter is a bit better because it depends on positive regulation from RAC, which is both a repressor and an activator. These simple control systems have some level of basal expression of your protein, even in the inactive state, and that small, unwanted level of protein expression may cause metabolic burden, resulting in some mutant bacteria that do not produce your protein. By the way, I have detailed videos on these operons linked in description in case you're interested. Let's attempt to establish a tighter control so that the leak is kept to an absolute minimum. This is the case where you switch from bacterial promoters to viral promoters like T7. These vectors are called pET vectors, which stands for expression by T7 RNA polymerase. To use these vectors, you put them in a bacteria that is engineered to express the viral T7 RNA polymerase. The T7 polymerase is typically under the control of a LacP promoter, regulated by the LacP operator. This strain of bacteria, technically a lysogen, is the DE3 derivative of BL21. The names are not important. It was made back in 1980s and it is still used today. The bacterial genome and pET vector both make the inhibitor that keeps all operators off. Now we have inactivation at two levels. The first level is on the T7 RNA polymerase gene, and the second is on the operator on the vector. Since this system depends on negative regulation by LacI, the bacteria will make a tiny amount of T7 RNA polymerase that'll cause a very minor leak in expression from the vector. This leak starts from the T7 RNA polymerase, and because there are two levels of inhibition, the leak is smaller than the simple system that we saw earlier. Since this system also depends on LacI inhibitor, you use IPTG to induce the expression when required. For most applications, this minor leak is fine. But if you want to express some toxic protein, then even one toxic protein molecule may kill the bacteria. So let's aim for an absolute control and absolutely no leak in protein expression. If we could somehow kill this small amount of T7 RNA polymerase, then we will not have any leaks. Back in 1990s, a plasmid carrying lysozyme gene was introduced in this BL21DE3 bacteria. This is a low-copy plasmid, and you usually see the S and E versions of it, and the bacteria strain names get the correct suffix accordingly. I will discuss the S and E plasmids at the end, but regardless of your choice of plasmid, this produces the lysozyme protein, which inhibits the T7 RNA polymerase. Now, even if you have a leak in T7 RNA polymerase, there's no leak in the protein expression from the vector because the RNA polymerase is inhibited. Now, let's induce the protein expression. You add IPTG, which inactivates the inhibitor. It cannot bind the operator anymore. The T7 RNA polymerase expression goes up, which can bind the T7 specific promoter on the vector to make RNA, which means you see induction of your protein. For comparison, by and large, T7 promoter systems give you the highest protein yield. Okay, back to this activation scheme. The lysozyme is still around. It is always present in the cell. It is independent of induction and always expressed. And this small amount inhibits a small amount of T7 RNA polymerase, even in the induction phase. But you make so much more T7 RNA polymerase that this tiny bit of inhibition does not matter. There's actually something very interesting about how this small amount of lysozyme is made. Let's discuss the silent and expressed lysozyme plasmids. In T7 phage vocabulary, the lysozyme is called gene 3.5. In both the lysozyme plasmid, the gene is cloned in front of a tetracycline promoter. In the E, or expressed version of the plasmid, the 3.5 ORF is cloned in the direction of the TET promoter. In silent plasmid, the ORF is reversed. So if you try to express the lysozyme gene using the TET promoter, it doesn't work in the silent version because the ORF is opposing the promoter, hence the name silent. The orientation works in the other one, so it is called expressed. But in pET expression system, this promoter is not used. So how does it work? Well, in this cloning, the 3.5 gene fragment wasn't the only thing present. In front of 3.5 gene is the T7 promoter for the next viral gene, gene 3.8. In this fragment, the 3.8 ORF is cut off, but its T7 RNA polymerase specific promoter is still present. In both these plasmids, the promoter comes after the lysozyme gene. So in this system, whenever you have T7 RNA polymerase around, the polymerase binds to the plasmid, converts the entire plasmid into a giant mRNA. Since this promoter and lysozyme genes face the same direction, a sub-portion of this giant mRNA is a lysozyme ORF, which gets translated and lysozyme protein is made. So, in a way, the T7 RNA polymerase positively regulates the lysozyme production, but lysozyme inhibits the polymerase. So, it is autoinhibitory, or this is a negative feedback loop. I hope this gives you some idea about controlling protein expression.